Section 6.6, Meiosis and Genetic Variation. Sexually reproducing organisms have a distinct advantage over asexually reproducing organisms. Number one is they create genetically diverse offspring. Okay, because of this variation or genetic diversity, they respond to changes in environment differently. Some having the ability to survive and in turn reproduce, while others won't be able to adapt to the environment and they will die off. Three factors that contribute to genetic diversity are: number one, crossing over; number two, independent assortment; and number three, random fertilization. And we'll talk about all three of these in the upcoming slides. Crossing over is when homologous chromosomes exchange genetic material. Now this happens during prophase 1 of meiosis, and what it does is creates unique genetic combinations. This is called recombination or genetic recombination. It's a mixing of alleles, mixing from the maternal and paternal chromosomes, resulting in a new genetic combination. So as you can see, these guys are crossing over. And it happens in such a way where it will actually break at this section, and then they'll reattach. The red chromosome will attach here, and this one will attach here, and it's making it a unique new chromosome. Linked genes are genes that stay together during crossing over. Linked genes are located close to one another on a chromosome, and as such, they wouldn't follow the law of independent assortment. So as you can see, we have these four genes, 1, 2, 3, and 4. 1 and 4 are the furthest apart, so they would most likely be involved in crossing over with their homologous chromosome. Gene 2 and 3, these guys are very close together, so the likelihood that they would be separated during crossing over is very, very low. So since they're close together and they won't get involved in crossing over, they're said to be linked genes. Independent assortment refers to how homologous chromosomes can line up in one of two ways for each set of chromosomes during metaphase of meiosis. So they could either go moms, which we're going to put in red, with dad, or dads with moms. It's random for each pair. So for the 23 different pairs of chromosomes we have in humans, that would give us 2 to the 23rd different possibilities of how they would line up, creating 2 to the 23rd different, genetically different types of gametes formed during meiosis. So here's a easy little example of how independent assortment would happen in meiosis. So with this, we only have two chromosomes. So these would be the paternal dads. We said those are in blue. And these would be the maternal. And in this case, they both happen to be, all the moms are on one side and all the dads are on the other. So when they separate during the meiotic divisions, you can see down here we get this combination of alleles formed in the gametes, or chromosomes found in the gametes. In this case, what we're getting is one of moms going to go this way, and the other of moms going this way, and this one would be dad, and this would be dad. And as you see, when they separate, we're getting different combinations of gametes formed during the second meiotic division, or to form the gametes. The last factor that contributes to genetic variation in sexually reproducing organisms is random fertilization. So it's completely random which sperm cell is going to fertilize which randomly released egg cell during sexual reproduction. If there are 2 to the 23rd different genetic combinations in sperm cells in humans, and 2 to the 23rd different genetic combinations that can be produced in egg cells, and the probability of one sperm randomly fertilizing that one A randomly released egg creates about 64 trillion different genetic possibilities in that offspring.